or if you just
Well,
want to plow into introducing Christian. Either way, you can.
We finished up the first class, the second session of the first class last night. Again, I want to big shout out to the people who attended and did a really nice job on doing their homework and IDing mushrooms correctly. So there were questions last night for when we're doing it again. And I'm going to kind of wait and see. It's really gotten dry, especially with that heavy wind we had yesterday. Supposed to rain. The forecast I'm seeing is supposed to rain every day next week. Those forecasts are notorious for changing as we get there. But if in fact we do get some more rain, I'll check and see if and when the mushrooms start coming up. And yeah, I'd be good to do another class.
Nice. Oh, then two more things. I forgot Margaret was there too. I didn't know if there was anything that Margaret wanted to chime in about. No, okay. And then lastly, yeah, it really sucks to be isolated, but if you're local especially, and want to really get involved, the web definitely needs more attention to it, as well as other positions. People are, we've been doing it for a while, and some of us want to freshen it up or take a break or something, and we're more than willing to hang out and get to know people and show them how it's done, and ease people into assuming these positions and stuff, and it's all good, and it's a lot of fun. You learn a lot of stuff. I suggest trying it. You can always say, hey, this doesn't work for me, and then what you're getting yourself into. But then influence others. Say, hey, there was this cool group, they hung out, they did stuff online, help them do stuff, because it's all about getting what you want to see back out of it, and we need help to do that. Lots of perspective, lots of hands, good stuff. Anyway, Phil, back to you.
Oh, we're ready to introduce our speaker.
Yeah, we're good.
Cool. So I've had the good fortune of knowing Christian from about 5 days after he came to Santa Cruz, and ever since then, being able to work with him, doing forays together, to drinking beer together, and it's been a pleasure to watch him develop into the mycologist, the professional mycologist that I see him today. So we hope that we can continue to work together, and once this COVID crap gets over, to go drink more beer. So ladies and gentlemen, let's welcome Christian Schwarz.
There he is.
Hi, everybody. I want to say especially hello, well, to all of you who I haven't really gotten to see in person for so long, which is very weird because normally I see all of you at least quite a few times during the mushroom season. So it's nice to be seeing all your faces. I have one of my current students in this class, or in this Zoom meeting, which is cool. Hi, Michael, at least one. And also one of my former students, Tiffany. Hi, Tiffany. Really good to see you.
Really good to see you.
Yeah. I just, a few weeks ago, started teaching my UCSC class again, which is nice. It's obviously very strange this year because we're doing it all remotely, but they're doing a really good job of showing up for classes and engaging and getting out in the woods on their own and seeing the mushrooms that normally I would be seeing with them. So it's really a little bit tragic that we're sort of having to do a field class not in the field, but they're making it work, and I'm really grateful to them for that. So I'm glad we're making it happen. Okay, so tonight I'm going to be talking about a project that I've been working on since 2018. Is that right? Yeah, I think that's right. And it was funded mostly by I applied for a grant from the Save the Redwoods League, which is a local nonprofit that specifically focuses on redwood conservation and issues around the conservation of that one tree. But as we know, redwoods are not just a tree, they're an ecosystem, they're a habitat type that has a lot of other organisms that live with them. And I sort of applied for this grant on a whim. I didn't know if there'd be any sort of interest from their organization in research about fungi, but I noticed that they hadn't ever, looking through their list of grants that they'd given out before, I noticed that they hadn't ever given a grant to anything related to fungi, or at least none recently, and certainly not about macrofungi. There might have been one related to some foliar endophytes or root endophytes, but there was nothing really about mushrooms of the redwood habitat. So I decided to just write a proposal and to try it. And much to my surprise, they said yes, and they decided to fund it. And what they funded was basically 3 years of monitoring, of long-term surveys of redwood-associated macrofungi. And I'm not going to say too much more right now. I'm just going to actually start this presentation because I'm going to talk more about what long-term monitoring even is in my talk. So I'm going to share my screen now. Hopefully I can do that. Yes. Okay. So you should all be able to see my screen now. Everyone, thumbs up. Yeah. Okay. So the talk is called Long-Term Lessons, Perspectives from 3 Years of Macrofungi Monitoring, or monitoring macrofungi. So here we go. What does LTMM stand for? You're going to see this abbreviation a few times throughout the talk, and it does not, in fact, stand for lettuce, tomatoes, mayonnaise, and mustard, but rather long-term monitoring of macrofungi. And the first question, we sort of understand what macrofungi are, and we sort of understand intuitively what monitoring is, although I'm going to have to explain that. And the first thing I should start with is the phrase long-term. 3 years obviously is not truly long-term in an ecological sense. This is the blink of an eye. But what I mean by long-term is surveying the same locations over a multi-year, or ideally, if you can get away with it, multi-decadal or indefinite duration. So setting up monitoring that goes on for a long time or longer than normal, visiting the same locations in a sort of concentrated way is what this project was all about. But there was a few additional things added on top of that. It wouldn't just be visiting the same locations every year once. My idea was to visit the same locations frequently and regularly, which means every 7 to 10 days or so during the mushroom season, as much as possible. Obviously, it's not always possible. Roads close, roads wash out, scheduling gets weird. But the idea was to visit these locations as regular or as frequently as possible, so that you would be able to detect shifts in what was fruiting at any given time, and you'd be able to see things start to fruit and mature and end. So I thought that 7 to 10 days was a feasible interval to do. And then the idea was also that I would quantify my effort. And this sounds fancy, but it's really not. It's just a way of expressing that you went to a limited area and spent a certain amount of time there. So I keep track of when I arrive at my transects, which I'll talk about more in a second, and I keep track of when I end those transects, so that I'm saying I spent an hour and a half covering this small area, so that in the future, people can compare how much effort it takes to detect similar amounts of diversity or similar amounts of volume of fruit bodies. So all of that might sound sort of confusing, but it's really very straightforward. It's just keeping track of the area you surveyed and then your start and end times of when you're surveying. So my protocol was that I would establish a bunch of 1 kilometer long transects, and that really just means a trail. A transect in this sense is just a start point and an end point that you walk from start to finish, out and back. So I end up doing 2 kilometers of walking for every survey or every transect that I survey, every visit. I record my start time and my end time, as I already mentioned, which helps to standardize the amount of effort that I put into it. And then during that time, I record all the species that are present on that transect, and I count the abundance of each species. So that's something that is sort of fundamentally different from a lot of other efforts that have been done on macrofungi in years prior. Is people would do presence or absence lists, where they would just list the species they saw, but that doesn't tell you if you saw 100 of them or 1,000 of them or a single one of them. And obviously that matters. Those are different biological phenomena if a mushroom is only producing 1 fruit body versus 100 fruit bodies. And as we all know, it's not easy to tell one individual from another, because one individual may produce many fruit bodies and multiple individuals may overlap in space. So this is not an attempt to count the abundance of populations by saying how many individuals there are, but rather how prolifically they're fruiting, how much biomass they're producing, how much sort of reproduction they're doing in a year. And it's sort of as a proxy. It's our best way of estimating population trends over time, because it's really not feasible to disentangle individuals from one another in the field. So I thought I would just set up as easy of a protocol as possible so that it would be possible for other people to pick up on this effort and do it after I can't anymore, or to start their own projects in other parts of the world, other parts of California, other parts of the United States, whatever they want. So it was intended to be a very low barrier to entry to do this protocol on your own. So here are my observations of fungi in Santa Cruz County from iNaturalist in the past 3 years. And as you can see, I have these sort of core areas that I've been going to. Most of my I haven't really been doing footloose and fancy-free, careless, fun mushrooming like I used to. I know a lot of you used that we would hang out in years prior, Phil and Dan and Justin. We would go wherever we felt like it on any given day. It would just be like, where is it fruiting? I don't know, let's go find out. And we would just go a different place every day. And that's really fun. And it's certainly the way to get familiar with mushrooms. But I started wondering, what do I miss out on by not forcing myself to go to the same places repeatedly, even though it's a little bit less fun. It's not as impulsive. You don't get to chase the rain around. But I suspected that I was missing things, that there was things I would learn if I started concentrating my efforts in smaller areas. So these 4 circles here encompass all 10 of the transects that I currently visit. And this is sort of what they look like. Here's just little maps showing the trails that I take. And these aren't all to scale. So on the left, there are the 3 different transects in Big Basin. I have one that's sort of a funny shape in Upper Fall Creek. I have 2 near Marshall Field, Twin Gates area, off of Empire Grade. And then I have 2 at UCSC, like pretty much on campus. And then I have 2 different transects at Nicene Marks. And so I visit all 10 of these transects approximately every 7 to 10 days. Now, in practice, it's not exact, but that's the sort of interval that I'm aiming at. Every 7 to 10 days, I visit all of these. And then I leave a gap, and then I do it again. And you might be asking yourself, okay, you're walking these trails and they're through forest. How do you know which mushrooms to count? Do you go off trail? If you're standing on the trail and you're seeing mushrooms, which ones do you count and which ones do you omit? And basically the idea was that I would use this. So if you take this graph on the screen as sort of an estimate of effort, how much time you spend at different distances away from the trail, approximately 95% of my effort is within 2 meters on either side of the trail. So these really are trail side surveys. I'm not trying to count every mushroom I see off in the forest away from the trail. I'm counting what's pretty much right next to the trail. But I do, if I see a big fruiting of mushrooms way out, 10 meters off the trail, and it seems like a very significant thing that's happening, I do want to capture that in my data. So I pretty much limit myself to 10 meters away from the trail. That's the furthest out I will record a mushroom, but I really spend very little of my time focused in that area because obviously you can't see a mycena 10 meters away very easily. So I'm really counting trail side mushrooms with a few inclusions of things that are further away from the trail if they seem sort of like a significantly large amount of fruit bodies. So what you do is you take this curve and you place it over different parts of the trail, and then you trace an effort curve around that trail. And so it's 1 kilometer long and approximately 2 meters on either side of the trail. Any mushrooms within that area, I'm counting. These are trail side surveys. I didn't want to make this protocol any more complicated. A lot of people who've done biological surveying know that in ecology and academic biology, there's a lot of randomization and standardization where you take a random number generator and a 1 by 1 meter quadrat and you throw it over your shoulder and you do a lot of voodoo basically to try and make your data statistically randomized so that you're not introducing bias. But I always found when I had tried to apply this to mushroom surveys that it didn't work as well as it does for botany, where there's sort of a greater density of plants in the landscape. So you're less likely to miss significant chunks of what's going on, a real picture of what's happening. So this is more of it's actually a known biological survey method called the controlled intuitive survey, where you limit your area, you standardize for your effort, and then you really just do what you do as a mushroom hunter, which is to look for all the mushrooms and count them. So I tried to make this easy on myself and easy to replicate for future workers. So like I said, I go to 10 transects throughout the county, mostly on Ben Lomond Mountain, plus the 2 in Nicene Marks, which are not on Ben Lomond Mountain. And I visit them throughout the mushroom season, approximately October to May. I try and start before the rains begin so that I get some zero data where there's really no mushrooms there and I can show that there's very few mushrooms. And then I end whenever the rainy season has dried out for about a month and the mushrooms start to decline and I get fewer than 20 species per transect. Once it drops below that, I kind of quit surveying. So this ends up being 6 months of the year or so. And this, I'm still in my 3rd year, so I'm halfway through my 3rd season of this. So I will have more to report in the future when I do some analysis of this data, but this talk tonight is just about some of the preliminary data that's come out of this, just so you can get a sense of what I've been learning out there. So once this is all said and done, I will have walked about 800 kilometers and spent about 500 hours of direct field surveying and generated something like 11,000 occurrence records that are attached to quantified effort and abundance counts of the species that I've encountered, which allows for a lot of really interesting fine-grained analysis on different patterns that I never would have had enough data to analyze just doing scattershot mushroom hunting like I used to. And believe me, I want to get back to that at some point. I'm not going to do this forever, but I'm really glad that I have done this so far. I've learned a lot. So this is what a notebook looks like on a busy day. This is one transect on the Upper UC campus. This wasn't even at the end of the day, but at some point it struck me while I was surveying this transect that it was having a really good day. This was actually just a couple of weeks ago. It looks like 20th of January. By the end of this transect, I'd spent about an hour and 45 minutes and I had detected 91 species in 1 kilometer of trail. So that's a lot of mushrooms to be in one kilometer. This is a very rich transect. So when you start to add this all up and analyze the data, and this is really just data from my 1st year of surveying, this is going to look different once this is all done. But this is just a graph comparing species diversity on all my transects. None of them had fewer than 330 species. So every transect of at least 1 kilometer length that I established in Redwood forest or Redwood dominated forest had at least 300 species on it. The difference between the most diverse transect, which is my Nicene Marks North transect, and the least diverse transect, which is the, I think now it's actually the UC Santa Cruz Central transect, which is between buildings. So it's very urbanized, very impacted, very disturbed. It still had 330 species on it or so. So it's about 100 species difference between the most diverse and the least diverse transect. But they were all very diverse by most accounts. So on my most diverse, or sorry, overall, if you add all the transects together, I have about 550 species represented now. So that's a large chunk of the known diversity of Santa Cruz County's macrofungi, just in 10 1 kilometer transects, which suggests that we actually have way more mushrooms to find in Santa Cruz before we saturate our species curve of all of the macrofungi in Santa Cruz County. We're not even close, I think, to knowing how many fungi really live here. And so I just want to sort of profile some of the fun stuff, the disjunct, the rare, the undescribed. These are sort of the flashy results. what I mean by disjunct is, for those of you who don't know that word, that means species that occur here that are more common elsewhere in the world. And this may be the southernmost known part of their range, or it's sort of disjunct just means separate from the main population of the species. And then rare, that's self-explanatory. That just means species that are not at all common. They may be globally rare. There's nowhere where you can go to easily find them anywhere. And then there's the undescribed species, which actually isn't even that interesting. As you all know, a lot of mushrooms in California remain undescribed. And if you're not finding undescribed mushrooms, you're not doing accurate transect surveys, because there are lots of them to be found, and you should be finding them, because unfortunately, a lot of our mushrooms remain unnamed. So,
Christian.
Yeah, go ahead.
Quick question. Do you want to take questions as they come into the chat, or would you like to get them at the end?
I actually don't mind taking a few as they come into the chat. If you think they're a good question, Dan, feel free to read them out.
Oh, okay.
Or you can, I mean, I just can't see the chat window, so I'm not going to be reading the chat messages as they come.
Right, I understand that. That's why I'm asking. Well, you have, well, one was already answered about how far you go on either side of the transect. How long does it typically take to walk your 1 kilometer transect?
That's a really good question. So when it's the beginning of the season and there's not much in the way of mushrooms fruiting, it can take me 30 minutes, 45 minutes to just go out and basically record a bunch of zeros, 1 or 2 polypores, a few little wood rotters maybe. There's almost always something happening. Even in the middle of the summer, there's mushrooms. But sometimes it can be pretty quick. It's sort of a brisk walk. But then when it gets really slow in the middle of the winter, in December and January, when there's just tons of mushrooms and there's big abundances, so lots of little mycenas that I have to stop and estimate how many there are in any given patch, it'll take me 2 hours or more to finish a transect. So it really varies. And that's why it's important to standardize your effort by recording your start time and your end time. Because obviously, if I ran a transect at top speed, it wouldn't be very good data collection. It would indicate that I'd done it too quickly. So that's why I standardize my time and effort.
Okay, and you also had a question. Do you count the hard brackets that stay there year round? How do you deal with that type of function?
Who asked that?
You can probably guess.
I knew it was
Tiffany.
That's a super good question. So the reason that's a good question is that it isn't intuitive what the answer should be. Because at first I was like, okay, I'm monitoring dynamics of macrofungi, things that are showing up and going away. But then I realized perennial polypores, they're dynamic too. They're just dynamic on a scale that I'm not very sensitive to, because they may be decade-long dynamics. Yeah. They're longer than sort of human attention typically sort of centers itself around. And so as soon as I realized that, I was like, of course I should be recording these things. So I record all the perennial stuff too. Okay. And that is funny, because you arrive at a transect and you know, I'm going to see those again today. Like you can write down, oh, I know there's 4 Ganoderma at the start of this transect, and you write those down every time you go. And as you'll see later, I'm really glad that I decided that I didn't just focus on fleshy macrofungi.
And I guess you could measure them periodically.
Yeah, but another reason is that those dynamics that seem long, like decades long, you don't know where in the decade you are. And that mushroom might go away next week. And it ended its 50-year lifespan during the course of your surveys. Or fire is a whole nother issue, and we'll talk about that later. But yeah, that's an awesome question, and I decided that I am going to record all of those.
Okay, good.
What is a much harder question to answer, though, is what do I do about the sort of in-between species that are persistent but not perennial? So their fruit bodies are still present, but they're not actively sporulating. So things like Sterium that dry out and quit sporulating, or Tricaptum, where their fruit bodies are all dried in place and they're still present, but they're not actively churning out spores. And what I ended up deciding about those is that I would record them if I thought they were fresh enough to be doing active ballistospory, which is shooting off spores actively, because I wanted to capture those dynamics in the less persistent species. So the Ganodermas are pretty much always sporulating. They do have cycles, but I can't really detect them. It's very hard. But the stereum, you can actually tell when they're fresh or when they're dry, more or less. All of this data collection is I'm doing the best I can. You know, there's no way to do this at scale perfectly. So I had to make subjective decisions about what kind of data I wanted, and I just ran with it from there. And that's going to be something that's going to have to be updated and changed and worked with as more and more people try this protocol out in different places. It's, you know, I made this protocol up and it may not be perfect. There's probably lots of room for improvement.
Okay, and finally, okay, do you know all these on site, or do you occasionally take samples or sequence to find out the ID?
Really good question. I know
Tiffany again.
a lot of them on site, and you actually kind of have to know at least a good chunk of them on site, or else your data collection just becomes impossible. You're like stopping every 10 minutes to be like, what is that thing? But that said, I've run into lots of things I can't identify. And there's lots of things I don't even try to identify. So for some groups, I'm only recording genus level data because they're just too difficult to identify in the field. And I would just like to capture their population abundance more than species level abundance. So for things like Leptonia, where it's just not feasible to do every collection to species, I lump those under one genus. And if there's obvious subgeneric differentiations, like for Cortinarius, I'll split out the Telemonias from the Phlegmasiums. So I always do it to the finest level that is practical, but in some cases it's genus level, which is, in the future, as people get better at identifying mushrooms, as we build more knowledge, we'll be able to do more of them at species level. But right now, I do what I can, and that's all we ever can do, right? I can't do more than I can.
Okay, great.
All right, I'm gonna get back into the talk now. So here's an example of a cool disjunct species. This is Hayderia abiatus, and it's quite common up in Northern California and in the mountains. It grows on little Douglas fir needles. It's an ascomycete that looks kind of like a little matchstick. It's got a fuzzy stem, a little hairy stem. And this appears to be more or less the southernmost limit of its known distribution in California, which isn't too surprising. This was at Big Basin, sort of in the core, wettest Douglas fir redwood forest. And I've noticed that further south, as Douglas fir becomes more and more a species of drier, open habitats, you get less conducive conditions for a mushroom like this. So this is just a cool sort of northern species that was present on one of my transects. And I'm really just doing a show and tell for these next few slides of some interesting fungi that are on my transects. Lepiota rhodophylla. So this is one of California's rarest mushrooms. And since I know Noah's in the audience and he asked a question about this on Facebook a few days ago, it's not totally 100% certain that this represents the same species as Lepiota rhodophylla. I have a collection of it. So this is an example of something that I found on the transect, and I also vouchered part of it because I wanted to get a DNA sequence. But so far, the microscopy, more or less the gross macromorphology, lines up with what is known about Lepiota rhodophylla, which was described from a single patch of cypresses in San Mateo County, and until now was not known from anywhere else in the world. So it's quite a rare species that is currently listed by the IUCN as critically endangered. And this was at one of the Nicene Marks transects. I don't think I ever would have found it if I wasn't doing repeat visits, because it has a short fruiting window. It came up and I hadn't seen it at the transect before, and I didn't see it the survey after. So I had that sort of 1 week long window where I detected it, and that was it. Thankfully, I saw it again this year. So it's nice to know that the mycelium there is persistent. Here's an example of a very drab looking mushroom. You wouldn't think a lot of it just walking by it, but because it was on my transect, I was forced to pay attention to it and to spend a little more time with it. And that's one of the sort of benefits of these transects is anything that's on the transect becomes important to you as the data recorder, and you sort of take a responsibility to figure out what it is if you don't know it, because you're taking sort of a brief ownership of the knowledge of these transects during the time that you're doing them. So I spent some extra time doing this, and actually the class that Tiffany was a part of did sequencing on this mushroom. These mushrooms that are in this picture were sequenced by my students at UCSC, and it turned out to be a representative of this genus Mycinella, which I had never heard of at the time. And it turns out is really poorly known from the United States. And this is so far, I think, one of the only records for California that's confirmed. I'm sure that it exists quite a few places elsewhere in the state, and people are just overlooking them or misidentifying them. But we know that it exists in Santa Cruz County now. And whether it's a known species remains to be seen. Thankfully, I also found this again this year, which is especially surprising given that it was in one of the burned areas. And I wasn't sure if it was going to survive that dramatic habitat shift after the CZU fire, but it's still there. It hung on through the fire and it's still fruiting this year. Another species that actually, some of you may remember the roaches, Pat and Courtney. They've moved since, but they were members of the Fed for quite a while. And they actually introduced me to this mushroom. They found it up near their place in Boulder Creek, and that's actually the collection that you see photographed here. And it's in this genus that is quite poorly known in North America. It's in the genus Trichosabe, which just means hairy head. And as you can see, it's not so obvious in this photo, but in these next photos, you can see that it has these little fibers all over the young caps. It's a white spored mushroom that grows out of wood. So it's sort of hard to place in any other group. At first, maybe there was some thought it was a Callistosporium, but it's not very brightly colored, and it's quite small, which some Callistosporium, not Callistosporium, Tricholamopsis is the genus I was thinking of. But yeah, this is present actually on multiple of my transects, this Trichosabe species. And we're trying now to figure out whether or not it's a described species. There's one known, Trichosabe puberula, which is known, and it looks pretty similar to this. But yeah, we'll find out soon, hopefully. I think we even have sequence data for it already. Okay, but that's just sort of the flashy results, the undescribed species, the disjunct species, the rare species, the stuff that I really wanted to do these transects about, because you can find all these species just doing scattershot mushrooming for fun. But what I really wanted to know was a better sense of how mushrooms behaved over time and through space. So the seasonal progression, I wanted to get a finer picture of that. How long does a fruit body last, an individual fruit body? And I found out some cool things about that as well. So I'm going to talk about those now. So I think some of you who watched one of my talks last year for the Fed have already heard this story, but here's an example of a mushroom that is actually in itself interesting just in the fact that it occurs in the county. This is Gymnopolis punctifolius, and we're pretty close to the southern range limit of this species. I don't know if it's ever been recorded in Monterey County. It seems possible that it would grow there, especially in the wetter, older growth forests. But Santa Cruz, it's rare here even. I only know of 2 locations for it, in Big Basin and at Nicene Marks. But I found one fruit body of it on one of my transects in Big Basin. And I thought it was so cool that it was there that I just sort of took a particular mental note of which log it was on. And then every transect or every survey that I made on that transect that year, that season, I made an effort to check on that one individual fruit body to see if it was still there, because you know, it's a rare species for our area, and I just wanted to get it on my transects. And much to my surprise, it kept being there. It kept being on my transects. And by the end of the season, I went back, and on the day that it disappeared, the day that I didn't see it anymore, I went back and looked through my notebook to the first day that I had seen it, and the dates in my notebook show that it was present for a minimum of 105 days. One fruit body, which is not at all what I expected from a gilled mushroom. I think of them as being sort of week-long occurrence or 2-week occurrence, but some of these species, and it's not all of them, most mushrooms are in fact 1 week or 2 week persistent, but this particular species is really rot resistant. It doesn't decay rapidly. And the data in my notebook was enough to show that this one fruit body lasted a minimum of 105 days. So it's just a really cool example of the kind of data you get when you revisit an individual area over a long period of time. It changes how you think about the ecology and natural history of these organisms. Okay, so then diversity through the season. This is sort of a different level of pattern that I'm looking at here. But if you take an average season from October through April, and you plot the time progression through the season on the x-axis and the number of species on a transect on the y-axis, if you plot all 10 transects, this is my first year of data, you get what you'd expect. There is such a thing as a mushroom season. You start with very low numbers in October, and then in January you peak and you get hundreds of species on a transect. And then it drops back down to 15 or 16 or below 20 species by the end of the season. But being able to this is 1 year of data or one season's worth of data, by making these sort of graphs for these 10 transects where I surveyed them in a very rigorous, standardized way, you can compare years in a way that you would never have been able to otherwise, or I would never have been able to otherwise. And what we're starting to see is a narrowing of this curve. So maybe in decades past, you would have a nice big fall season and then a winter season, and then you'd have sort of a spring season for mushrooms. And what we're seeing now is a compression on both sides, where the spring mushrooms and the fall mushrooms are getting closer to each other in the calendar year, and the season is just sort of jumbled phenologically. Another thing that we're seeing in some years, and every year is different, obviously, so this is going to take a long term building of this data set to really be able to say these things with any confidence. But in some years, we're seeing a bimodal distribution where there's two humps, a fall season, and then, like, for example, right now, January dries out and you get actually a dip in the middle of the season. And then there's another burst of rain in the spring. And being able to compare what sorts of mushrooms respond in different patterns, different kinds of years, is something that we don't have data about. And I think is just a really fruitful set of questions to look at, or set of patterns to explore and mine for future research. And now I just want, this is sort of a silly way of phrasing this, but it's an easy way. So a lot of things in nature can be viewed through an elemental lens, the effects of water, of earth, of wind, and of fire. And water and earth are actually very easy to talk about with mushrooms. Almost all of you already sort of know what I'm about to say. Rainfall is the single greatest determinant of diversity and abundance on any given survey. No surprise there. Any transect you go on, any survey that you do, if it has rained, you're likely to see mushrooms. And if it has not rained, if it's dry, you're not likely to see mushrooms. It's actually likely that you won't see very many at all. But of course, rain is not the only determinant. You can rain 15 inches in the middle of downtown LA, and that's not mushroom habitat because it doesn't have the right earth, doesn't have the right soil, doesn't have the right habitat. So habitat characteristics are the second most important determinant of diversity and abundance on any given survey. So if you're in really marginal, urbanized, impacted UCSC central campus type habitat, there's going to be fewer mushrooms than on the upper campus transect that's right next to it, just because of the habitat type, the sort of amount of soil that's available, and the quality of soil, and the sort of the richness of plant diversity that structures what mushrooms are there. So this is really no surprise. Water and soil, water and habitat are the most important structuring elements in mushroom diversity and abundance. But what I was really surprised to learn was just how much wind and fire have to do with mushroom diversity. Obviously, it's not surprising that fire impacts mushrooms, but I had never really experienced it in coastal redwood forest. So I'll get into that in a second. But wind is something I want to talk about really quickly. And it's very topical because, as you all know, those of you who live in Santa Cruz, the past week was a really windy one. We had howling gales a couple nights ago that were moving my house around. And I should show you, I was out doing a survey transect today, and there was so much redwood branch litter on the transect. It looked like, like the trees had shed just a large percentage of their branches down onto the forest floor. And wind really has an impact in, like I just mentioned, the availability of small diameter wood debris in the forest, which a lot of mushrooms use as their main substrate. So small diameter wood debris is made available by wind. So wind, sort of the regime of wind, how strong the winds are, how long they last, has a big downstream consequence for wood decay macrofungi. But the wind itself, just the air rushing through the understory of the forest, dries out fruit bodies in a way that's very notable for anyone who has experienced it when you're mushrooming, trying to collect fresh fruit bodies. But it has a huge selective force on the kinds of fruit bodies that mushrooms make. So there's a whole group of mushrooms that may be present in numbers that are up in the thousands or tens of thousands on one day, and then 2 days later, there will be no actively sporulating fruit bodies left if there's been a big wind event. And these are things like mycenas and gymnopus and macrotifula, things that have little tiny fruit bodies where each one has so much surface area and so little tissue that it just dries out in a snap like that. So there's this guild of mushrooms, like for example, here you see Mycena galopus, where it's not uncommon for me to record more than 10,000 fruit bodies on a transect on any given day. But then if I revisit it 1 week later, or even just a few days later, they may be down in the hundreds or almost down to zero if we've had enough wind. Here's just a close-up of what that mushroom looks like. You can see why they dry out so easily. There's almost no tissue there. They just turn into herbarium specimens in an hour. Macrotifula is another example of this, where they can just carpet the forest floor, especially in redwood forests in the early season when it's quite damp and the duff is really humid and the air is really humid. But their fruit bodies are just tiny. It's about as reduced as a mushroom fruit body can be. They're really just a noodle, a little spindle, a tiny bit of tissue sticking up in the air, and they produce some amount of spores on that fertile surface, but nowhere near as many as one individual fruit body of a bolete, for example. What they do is they make up for it by having many fruit bodies, producing many fruit bodies at once in one location. So their evolution of their fruit bodies and the timing of their fruit body production is probably really dramatically responsive to wind. It's probably been a major selective force in mushroom fruit body development, and it's something that I think would be interesting to do more research on in a more focused way. Obviously, my study here is very coarse, very general, very superficial. Pattern noticing is my goal. I'm not actually doing any sort of manipulative experimentation or hypothesis-driven research that you would expect from a more detailed ecology and evolutionary study. Okay, so that was wind. And now I want to talk more extensively about fire, because fire obviously has had a major, major impact on Santa Cruz County in the past year. So in 2020, the CZU fire started in the middle of the summer due to a big lightning storm event that was really rare for our area and very impactful. It started multiple small fires in the upper elevations of Ben Lomond Mountain. And then all the small fires merged and pushed west towards the coast and downslope as winds were pushing them. So wind actually amplifies the effects of fire. They're sort of the same thing when they really get going. So 86,000 acres, I think that figure is right, burned, most of it at pretty high intensity. This was not an understory burn. This was not a good fire in the way that land managers sometimes talk about it. This was an out of control fire driven by really historically low vegetation, moisture content, really high winds, poor management history where we had just let lots of forest grow dense, and there was just an accumulation of ladder fuels in places that it wouldn't have looked like that naturally, or at least not historically, when Native Americans were burning the landscape at regular intervals. And it burned almost all of Ben Lomond Mountain, at least, you know, the mid elevations up. Most of it was in mixed evergreen forest, as well as some chaparral, including the old growth redwoods of Big Basin, which is probably the most astonishing fact about the CZU fire, is that it burned a forest that I never thought I would live to see burn. 95% of Big Basin State Park burned at high intensity, and the fires were crown fires. They reached the upper parts of the canopy of redwood trees that are 150, 200 feet tall in some places. So this was just an incredible inferno. This is satellite view of the CZU fire aftermath, which shows just how much of the county was burned. 25% of the county's land area was burned. And it burned all the way to beach sand. You can see up there on the north coast, just south of the county line. It burned all the way to beach sand. I actually went there just a couple days after the fire, after we were allowed to go up the coast, and there were Monterey pines growing in beach sand that were completely burned. So interestingly, this is what it looks like in the upper parts of Big Basin State Park right now. Well, this is actually a few months ago. It looks a little different now, but you can see there was no litter left in the understory. All of the wood that was on the ground, all of the litter, all the duff burned away. It went down to a layer of ash on top of mineral soil, and all the trees are burned pretty much up to the canopy. Here's a sort of a even further into the old growth where the bigger trees are. This is a mix of tan oak, Douglas fir, and redwood in this picture. This was in November, and you can see that there's just a few stump sprouts coming off the tan oaks at this time of the year. And a lot of the redwoods pretty rapidly started doing what's called epicormic sprouting, where they produced new needles out of the trunks that were burned, because redwoods are very fire resilient. If they're able to make it through the fire, they'll start going again. But unfortunately, many of the redwoods didn't make it through the fire, and they actually burned so hot that they were killed. That said, there is significant amounts of redwood regeneration going on. So most of the redwoods survived. The Douglas fir did not. So the Douglas fir really can't survive high intensity fires, and they don't do epicormic sprouting. So there was extremely high Douglas fir mortality throughout Big Basin State Park. There was someone who studied Douglas fir in the aftermath of the 2008 Martin fire in Bonnie Doon. And this researcher found that any Douglas fir that had more than 60% of its canopy burned did not survive for within a year or two after the fire. And unfortunately, that applies to pretty much every Douglas fir in Big Basin. Almost all of them had greater than 60% canopy loss. So something is going to take their place, whether that will be mostly tan oak or some other habitat type, we'll see. And here's sort of what it looks like. So on the right of the screen, those are tan oak resprouts. I didn't know before this year just how good tan oak is at resprouting after a fire. But even though the upper parts of the tree may be dead, a lot of the stumps are still alive, and they're resprouting these little suckers. So the tan oaks will come back in some form. I don't know if they're going to attain the same sort of shape, the tall tree height, especially as the redwoods grow in and shade them out. So we may lose significant amounts of tan oak, not to mention that they're already stressed out by the presence of sudden oak death. So we're going to be experiencing lots of shifts in the composition of the tree community in the county in the aftermath of this fire. It really remains to be seen what the long-term trajectory of this fire does. But okay, what about the mushrooms? What has fire done to the mushrooms on these transects? So the CZU fire burned 4 of my transects, which is kind of amazing because I did not intend for this study to be a post-fire study. And this sort of major new dimension was added to my data sets because I just by pure luck managed to get the last 2 years of baseline data in a quantified way from Big Basin that we're going to get for the next 100 years. It's never going to look like that in our lifetimes again. So I feel extraordinarily lucky that I managed to start collecting data right before this fire, and it allows for direct comparison now. So I haven't even done a full year of post-fire survey work in these burned areas, but I'll tell you now just some of the first things that I've noticed in these burned transects. So 4 of my transects burned, including some of the richest ones, some of the most diverse ones, which is really sad in some ways. I suspect there are mushrooms that were disjunct or rare in the county that were present in Big Basin that are just not going to come back, at least not in our lifetime, because that ecological sort of apex community there was already a relict forest. There wasn't too much old growth, big redwood forest left in this part of the state. And that was sort of the crown jewel, one of the most diverse places for macrofungi in the county. and it's just totally different now. It's not the same. But I have learned some things that I didn't expect I would. I had no idea that Coloriza umbonata was such an interesting mushroom until this year. I'd always sort of felt like it was kind of boring. It's an interesting mushroom because it has that strange morphology, that long tail that you're all probably familiar with. But it turns out it also has an interesting ecology response to fire. So just for example, I so on my first 2 years of surveys, I'm just going to highlight one transect in particular from Big Basin. During the survey years of 1819 and 1920, I recorded it on 6 visits. So probably the middle of the season in both years. And it wasn't a very abundant mushroom on these transects. I recorded a low of 1 fruit body to a high of 7 fruit bodies on this transect, with an average of 2 fruit bodies per visit, which is low. That's not an abundant mushroom on these visits. But after the fire, in my first 2 or 3 visits after the fire, on the same transect, the story was completely different. So it burned in 2020, and in the first few survey visits after the fire, the first one I found 125 fruit bodies of Coloriza. So huge, tenfold, or yeah, no, more than that. Yeah, basically a tenfold increase. And then my last visit there, I found 493 fruit bodies of Coloriza. So it has jumped up by 70-fold. This mushroom has dramatically responded to fire in a positive way. It appears to really be stimulated by the effects of fire. And it's one of the only guild mushrooms on the landscape there right now. And there's just hundreds of them right alongside the trail. I have to stop and count them just because there's so many of them. So this is a mushroom that appears not to just be fire tolerant, but perhaps we could make an argument that this mushroom is indeed fire adapted, that it is a fire loving mushroom. And there's lots of other ecological hypotheses that I'm starting to develop about Coloriza. It's really a mushroom that's captivated my interest this year. Yeah, but I'll talk more about that in a future talk once we learn some more about it. But okay, so what else have I learned about fire? So here's just an example of what it looks like. This ring had, I think, 155 fruit bodies of Coloriza in this arc around a burned redwood. So it was just a really impressive fruiting, like nothing I'd ever seen for this species before. And then there's the obvious fire responders that we expect to see. Ascomycetes like this Anthracobia. There are just tens of thousands of these little fruit bodies across the charred soil throughout Big Basin and throughout Fall Creek. And they are heavy sporulators. So I was standing in a patch of these and the wind blew a little bit and gusted over the tops of the fruit bodies and the sunlight hit them and they just released a huge cloud of white spores. And it was a really cool moment to feel like this is what's supposed to happen in the post-fire ecology. These mushrooms are adapted to these conditions where they suddenly have a lot more airflow in the forest understory. And these little ascomycetes really capitalize on that by having air-triggered spore release or wind-triggered spore release. Polypores, some of them are doing really well because of all the down burned wood, some of which has undoubtedly been chemically modified by the fire and makes it more amenable to the fruiting of these polypores or even just the colonization of the wood. This one, Lentoporia carbonica, is not one that I really had much familiarity with before the fire, and now it's abundant in Big Basin, and it's one of the most common mushrooms there right now. It's on all these blackened Douglas firs and redwoods. It's a really big, cool, impressive polypore that forms sheets like 10 or 15 feet long in some places. It's a really spectacular mushroom. And actually, if you find it, it's really easy to tell with a microscope what it is. I mean, it has a very distinctive look to it, but it's amyloid. All the tissues are amyloid. So you can just take a little chunk of it and put a drop of Meltzer's on it. You actually don't even need a microscope. It'll just turn blue or dark blue-black. So it's a very, very distinctive species. And then this was easily the most interesting thing I have found on my transects. A couple weeks ago, I showed up to one of my transects and got out of the car, and the very first mushroom I found was this thing. And I just had no idea what it was. It doesn't look like anything I've ever seen. And I saw 10 of them, and I thought, wow, 10 mushrooms that I have no idea what they are. And I looked across the trail, and there was 10 more. And I looked down the trail, and there was 20 more. And I looked down the trail, and there was 50 more. And I looked down the trail past that, and there was 400 of them. And then I walked the whole transect, and I recorded more than 10,000 fruit bodies of this thing. And I have no idea what it is. So to be surrounded in a fire landscape where there's almost no mushrooms happening by what's functionally a UFO in such high volumes, I was just astounded. I had no idea what to make of it. So I collected a whole bunch of it and took it home and did some microscope work, and it didn't help. I still don't know what it is. It doesn't really fit into any genus that I know of. We tried a few on for size, like Lyophilum. It doesn't quite fit for that. It's missing some of the micro features we expect. It doesn't fit for a waxy cap, so it doesn't seem to be in the Hygrophoracea. It could be a Clitosebula or a Leucoinosobe, which are these sort of more obscure genera, but there are none that are known to be fire responders. And this thing is big. Like the clusters can, be 15 centimeters tall, 16 centimeters tall, and they grow in bunches of 30 to 50 caps. But they're also soft. They sort of look like a Lyophilum, but they're very soft and fragile. Their fruit bodies are very, they're almost like a ruderal species. They're just like made in volume, but there's very little investment in the fruit bodies. So what these things are doing, what they are, I have no idea. I literally put them in the mailbox to be sent off for DNA sequencing yesterday. So we should know a lot more about what they are and what they're related to in the next couple weeks. So I'm really looking forward to getting data back on this thing. For now, I'm just calling it the exuberant cinder cap because it's fruiting in such exuberant numbers, and it has this sort of ashy looking cap, and it comes up through the cinders in the forest. So I'll just show you a few more pictures of it. Here's another view of it. And before you ask, it has been present at all of the burn sites. So it's in Big Basin, it's at Fall Creek. A friend of mine sent me pictures of it from the Bonnie Doon Ecological Reserve, which also burned. So anywhere where there were, it seems most associated with tan oaks. Anywhere where there was tan oaks that burned in the CZU fire, this thing is fruiting in huge numbers right now.
Are you ready for some burn questions?
Yeah, sure.
Okay. From Maya, doesn't the redwood rooter grow on the roots of redwoods? Perhaps it's taking advantage of weakened trees or benefiting from the lack of competition post-fire.
Yeah, so to give away it's actually not even a hypothesis. I feel like I know now what redwood rooter is doing. I don't know why it hasn't been more obvious in the past, but I did a little literature review recently about what is known about the ecology of redwood rooters and the function of that long tail root. And we know that redwoods are not mycorrhizal and Coloryza is not mycorrhizal, so it's not associated with the roots in any sort of mutualistic way. And we don't expect that it's a root parasite. There's no evidence of that really. And it turns out what it's doing is that it's decomposing small diameter redwood branches. So branches of about this size or a little bigger. It's also on redwood stumps too. And it turns out that this is actually in the original description of the mushroom. The first person to ever describe this mushroom knew this. It's described as being on a redwood stump in the original description. But curiously, if you look at the literature since then, there has been a lot of uncertainty. People seem confused about what it was doing. But it's a specialist, small diameter redwood wood decomposer. It's adapted for redwood wood. And I think the reason that it's so stimulated by the fire is that its substrate wasn't really affected. So redwood branches are buried deep in the duff. And when that duff layer burned off, it suddenly was much easier for those mushrooms to sort of produce. It almost acted like as a casing layer. And they were sort of stimulated to fruit. So whereas most mushrooms lost a big chunk of their substrate in the forest floor because of the fire, Coloryza didn't, because its substrate is buried. These branchlets become covered in soil and duff accumulations over the years. And so they're actually quite deep down. And that's sort of what that tail root is about. They produce primordia way down in the soil and then push their caps up through the duff. And that's another reason that they're unbunded. So that's a morphological adaptation. That's not just random. They're pointed in order to push through that really thick redwood duff layer. So that's sort of my current view of Coloriza.
Okay, and your Agaricalis species, which I found, it had me completely stumped until I looked on iNaturalist and saw you had written the name Leucoinosobe, and I said, oh, okay, that's good. Then I looked up Leucoinosobe and said, well, nobody knows anything about it, and there's only a few species known, so that's not so good. So
no, so I don't think it's a Leucoinosobe anymore. That was my first guess, and it sort of worked for about a week. But then I did some more microscopy using some chemicals that I didn't have that were supposed to confirm that one way or the other, and it didn't look so good for Leucoinosobe anymore. So the records are there right now under that name as a very tentative first guess. And I think there's still some chance that that's what it is, but like you said, it's a very poorly known genus. So there's plenty of reason to think that we don't know every way that a Leucoinosobe can look. We'll see. I'll definitely keep you all posted when we get results back.
Okay, but that one, yeah, definitely had me stumped because, it does look a little bit like a waxy cap. It looks a little bit like a lot of things.
Where did you find it, Dan?
Probably right along one of your transects, so where I'm not allowed to hike, but in Fall Creek.
You know what you're talking about, yeah.
Yeah, I was checking out a big waste dump in there to see if that had been cleaned out, and actually the fire cleaned it out pretty well. I'll tell you about that offline sometime.
Yeah, that'd be great.
Let's see, any other questions? No, I think that covers it. Yeah.
Okay, cool. So I'm just going to give you another sort of ooh-ah picture. Here's just a picture of how abundant they are. And I know it's not easy to see the mushrooms in this photo, but this is an area of about 3 meters square. And this is the exuberant ash cap here, or the exuberant cinder cap. And in this photo, I actually counted every cap that was present in 3 meters square. And there's, I think, 294 fruit bodies in 3 square meters. And what's really amazing is that you take that density and you multiply it by acres and acres of the landscape. This mushroom goes across hillsides, just as far as the eye can see in some places. There's just so many of them. So it's really an astonishing mushroom. I can't wait to figure out what it is. So once again, before I end, I just want to say thank you to Save the Redwoods League for funding this, and I'm really excited to finish out my last year of surveys this year in a few months here, and start summarizing more of the data and exploring even more of the patterns in more detail. Now, in some ways, the hardest work begins. It's not fun walking around in the woods. I actually have to do more typing and data analysis and figuring out what all this means. But that's hopefully a talk that I'll be ready to give for you next fall. So thanks for having me tonight, and I can take questions for a little while now if y'all want.
Well done, Christian. Well done.
Thank you. So you guys, you actually don't have to chat. You can just unmute yourselves and ask questions directly if you feel like it.
How about him? Release the hounds.
Unless I've put you all to sleep.
I don't know. Zev totally recognizes your voice. She's been really,
Hi, Zev.
really confused.
Aww.
I'm jealous of all my friends with pets. I want a dog right now. I've always said that no Zoom meeting is complete until there's a dog on chat somewhere. So thank you for providing our Zoom dog.
So true.
Okay, well, if there's no more questions, you can, oh yeah, wait. Maya, do you have a question?
Hi,
Hello.
how's it going?
Good.
Great talk. Thank you so much. I learned a lot. I'm so excited about the post-fire work that you're doing. I'm also doing post-fire mycological research right now. And I'm curious if you have intentions to keep returning to these sites after your grant is over. Or next step is.
I can't afford to, basically. It takes so much time that the only way I was able to do it was through this grant. And it's possible that they could renew it, but even then, it's difficult for me to make it work. It's just not, it doesn't integrate with other work very well because it's so intensive. So by itself, the grant was not enough to really float me. So I probably won't be able to do it at this intensity in the future. But what I'm hoping is that I can integrate it into an internship with UCSC students that are doing sort of a citizen science class that I'll be teaching next spring. But the problem is that you have to be able to recognize at least a couple hundred mushrooms sort of on site, which is the big challenge. And one way I'm thinking about making it possible for people to do this in a more feasible way is to reduce the number of mushrooms to focus on down to a core set and just count, learn to recognize maybe 30 to 50 species that are really keystone species, and at least have other volunteers continue the surveys that way. But I would certainly hope that even if I can't do these in the next few years, I would certainly hope that I could get together some sort of funding to do a revisitation on a decade-long interval. So maybe 10 years from now, I could come back and do an intensive 3-year resurvey of all the same sites and see how things have changed. But I would love for there to be no data gap. I certainly would love it for someone to be visiting these sites and making it so that we have a continuity of data. But obviously there's time, finances, practicality, feasibility issues that I haven't yet figured out how to overcome.
Got it. Maybe so.
Hello.
There was a question from Mason Lee on the chat. Actually, Mason, you can unmute and ask directly if you'd like.
I'd prefer that, actually. But, oh, mic isn't working, so we have to read it.
Oh, okay.
Sure.
How do you think these mushrooms, agaricales, affect post-fire succession in the ecosystem?
That's a really good question. And it's probably something that Maya is thinking a lot about right now is how do these forests recover? What role do mushrooms play in this? And I'm honestly doing so much less detailed work than what I think Maya is doing. I'm really just documenting what's happening. I'm not trying to do much explanation or manipulation. I know Maya's been doing some like remediation and sort of how to sort of speed up recovery in some of these places, but I don't know, but what some sort of like natural history observations that I can make that seem sort of obviously involved in succession is that one, when you have a huge biomass of mushrooms in a post-burn area, one of the first things that happens is that insects go, oh goody, and there's just masses of invertebrates and insects that flood in, and what follows the insects are birds. And birds are carrying seeds in their droppings. So it's a way for seed dispersal to sort of arrive in these newly burned areas via this sort of stepwise, the mushrooms happen, which attract the insects, which attract the birds, which bring the seeds. So that seems like a sort of obvious thing that's happening in some places, especially Big Basin. There's all these insectivorous birds of open land that I didn't ever see at Big Basin before, like black phoebes, that I now see lots of right in the core of what used to be very dark, dense forest where I would rarely see black phoebes. So different birds are in different areas. Woodpeckers are totally flooding into these burned stands of conifers, because they really like the effects of fire on their drumming trees and their granary trees. And what they do when they drum into trees is they open infection sites into the wood for polypores and other wood decay fungi. So woodpeckers and polypore mushrooms have a well-known association in sort of facilitating each other's existence in forests. Another way that they are facilitating succession is that for those ectomycorrhizal fungi that sporulate in burned areas, they're providing the mycelium that new germinating plants need to recruit and grow in a vigorous way. So thank goodness there are still some ectomycorrhizal fungi that are alive in these burned areas. I saw some swillis already fruiting in the core of the CZU fire. So they're going to make it so that new conifer saplings can grow up in those places. But obviously, there's a whole set of fire follower fungi that fruit in the spring that have not yet fruited. So I'm expecting the next few months, February, March, April, to be big months for interesting, weird ascomycetes in the burns in the CZU.
Christian, I was wondering if any of those species of fungus would actually be able to help out secession. I know this kind of like goes off of the last question, but if any of those species that we're going to be seeing pretty soon are going to be helpful for the forest, and if there's any way that we can spread that as just while we're out there hunting for mushrooms.
Definitely. I think almost all of them are helpful in the succession. That's why that's sort of how the ecosystem is structured. What happens not for a reason, but as a integrated network of cause and effect and response and dynamic balance. So whatever is going to happen there has to happen there from the natural point of view. That's just what is required for the ecosystem to do its thing. As far as us helping it along, I tend to be pretty wary of attempting to like amplify what nature is doing, because I at least feel like we have sort of a bad track record of knowing how to dial that in. But we are going to need to think about how to mitigate the impacts of all of the tree work that's happening along the sides of the road. So in big basins, it's pretty heartbreaking to see anywhere where the trees are near the highway, they're felling and dragging enormous logs and disturbing the soil in a way that big basin has never experienced. So that's going to have a major impact on invasive plants, on the intact nature of the soil ecology. It's not going to be the same, and it's not, thankfully it's just the corridor along the highway, but that's bad enough for invasive plants. I mean, that has a major impact in the encroachment and invasion of invasive plants into what is otherwise a very closed, intact native forest ecosystem. But I don't know. I don't know how to try and put our fingers on the scales and tip the balance of what's going to happen. It seems difficult.
Oh, sorry.
Yeah, that'd probably be tough to predict.
Yeah. And
Thanks
it's
for talking
also
tonight.
fast moving. You know, fire is not it's sort of a rapid shift in an ecosystem. We're seeing type conversion of habitats from Douglas fir probably to chaparral in 5 years. You know, that's a really quick change. And it's hard to catch up to that at scale. You could figure out what to do, and even if you knew what was going to happen, it would be hard to do it at scale across the whole landscape. Like, it's just, there's so much.
Now, what is the progression after chaparral?
It sort of depends on how much rain you get in the
Yeah, I
next.
guess it depends on the weather, whether
Yeah.
that region can ever support a large forest again.
That's one of the big concerns, that this fire isn't just a fire. It's a fire in the context of something that was already happening, a larger, longer process, which was that our forests were drying out. And when a fire happened, maybe in prehistory, it was happening in a relatively stable rainfall regime, at least for a while. And this, I mean, if you clear out a forest and you dramatically reduce its capacity to hold moisture at the same time that the climate is warming and drying, you're going to have more oak trees, more tan oaks, fewer redwoods, fewer Douglas firs, more chaparral over time. So we're changing our habitats.
A couple thoughts about some of the things that you've been seeing. If those exuberant center caps or whatever you're calling them are everywhere, that means their mycelium is everywhere and just never really fruits.
Yeah, so that's the really big question, is what was this thing doing before the fire? Like,
Yeah.
I've been visiting, I mean, Justin and I have been going to that spot. I mean, Henry Young has been visiting that spot for probably decades. And he took me and Adam Riska and Justin there, and Noah has been there. And it was never there. I never saw that mushroom there. So I don't know where it was or what it was doing. Presumably the mycelium, like you mentioned, was down there doing something, waiting. And what its ecological role is still a bit mysterious to me, because it does seem to like tan oaks. I don't think it's a mycorrhizal species, but it does seem to have a habitat preference more for tan oak than for redwood, although it's quite mixed in there, so it's kind of hard to tell. I want to do some isotope analysis on the fruit bodies, which will help us determine what kind of carbon it's getting, whether it's getting carbon directly from the soil or from a plant host, on the off chance that it's a wood decay species versus a mycorrhizal species versus a soil species remains to be seen. But I don't know. It's a big mystery. It's really cool to think about.
Yeah, and then also with all that duff burned off, I guess the redwood rooters aren't really going to have roots anymore because they're going to be right next to the things they're growing off of.
I actually found yeah, so that's an interesting point. If their branch layer is more exposed, do they have the tail?
Yeah.
And the answer is no. Today I found a branch with the redwood rooter embedded in it with no tail. It was just emerging straight from the branch because it didn't need a tail. It didn't have to get up above anything.
Yeah. How are you, like I want to go into the burn area and like look around and help and stuff, but it's still, a lot of it's still closed. You have like permits from the?
Yeah, so it was actually very difficult to get access to Big Basin. They wanted me to have a ranger escort every time I went.
Wow.
And I said, you're going to annoy yourselves if you try and work with my schedule, because my schedule is unpredictable and very frequent, and your ranger is not going to like me so much. So I still have to, like, you know, it's behind a locked gate and I have to call in and out before I go, and I have to wear a hard hat and I have to, you know, follow a protocol. But there are places that you can get into the burn. If you want to like get in touch with me via email or text, I can give you some tips on how to see this ecology.
Okay, cool. Thank you.
Yeah, I guess one of the dangers of going into the burn area is dealing with trees falling over the road.
Yeah, there's a lot of trees falling in these areas right now, especially after the windstorm this week. So
I can stream that video.
I would recommend not going into the burns this week until the trees have had a little time to fall.
Okay. Yeah, I did go into one area that was officially closed, you know, in part of Fall Creek. There was something, you know, very specific that I wanted to look for, and but it's an area that's, you know, relatively safe and the roads have been cleared, and
Yep.
But there a few areas in Upper China Grade Road that I visited every year for the last, say, 15 years at the same time of the year, and kept kind of spotty records on what I see there. And I'd love to see what's coming up now, but very hard to get to.
Yeah, yeah. Well, we should talk, Dan, because if you have records and if you know these spots, it would be really worth trying to negotiate access for you to follow up.
Okay. Yeah.
Okay, folks.
Good.
Thanks for having me tonight. And yeah, you'll be hearing more from me about updates from the fires and just from the transects and the data in general from the long-term monitoring project. But until then, see you next time.
Thanks.
Good to see you,
Thanks,
Christian.
Christian.
Bye.
Yeah.
Bye.
Everybody.
Bye.
Good night. Be safe.
Good night, all.
Good night.
Be well.
So, turn off.
All right.
I want to say goodbye to Leah and Phil. And
Bye-bye.
bye, night, Lee.
Good night.
Good night, everyone.
Good night.
Say hi to Phyllis.
Oh, she's in bed already.
Okay.
Good night, everybody. Nice seeing y'all.
Okay. Good night.
Over to the red there.
Bye, Lee. Still there, kitty? I fell asleep.
Yeah, I've just stepped away.
Okay. I guess I better